Hi everyone, today I will present a research article titled Plastic Pollution Affects Ecosystem Process including Community Structure and Functional Traits in Large River by Nava et al. 2024, published in Water Research. Usually, when we hear about plastic pollution, we immediately think about oceans. But have we ever thought about what happens when plastics stay in rivers for a long period of time? So, this study investigates exactly that. So before I explain the study, I would like everyone to take a look at these two images. The image on the left shows rivers that contribute to global plastic pollution entering the ocean. Interestingly, Sungai Klang in Malaysia is also listed among them. So, image on the right present another perspective of the issue by showing how plastic waste is connected to global waste movement and management. From this example, we can see that plastic pollution is no longer only an ocean issue. Rivers also play an important role because they transport and accumulate plastic waste. But here's something interesting. What actually happens after plastic enters river ecosystem? Does it simply stay there? Over time, microorganisms begin colonizing the plastic surface and form what is known as the plastisphere. These plastisphere communities are important because they may influence several ecosystem processes including biodiversity, nutrient cycling, oxygen dynamics, ecosystem metabolism, and overall productivity. However, although plastic pollution has received increasing attention, few studies have evaluated how plastic affects ecosystem functioning in freshwater system. Therefore, this study was conducted to investigate how plastic-associated communities may affect ecosystem functioning in large rivers. So, after understanding why this issue matters, the next question becomes, what did the researcher actually want to find out? This study had three objectives. First, to identify whether plastic type or river condition influence microbial growth and community development on plastic surface. Second, to evaluate the effects of plastic-associated communities on ecosystem process, especially oxygen levels and nutrient cycling. And third, to examine the relationship between biodiversity, biomass, and ecosystem productivity in large rivers. This objective helped the researchers understand not only what grows on plastic, but also how it might affect the surrounding river ecosystem. To answer this objective, the study was conducted in the Lower Mekong Basin in Cambodia. Three river systems were selected: Basap, Mekong, and Tonle Sap. The researcher incubated 4 plastic polymers for 30 days: polyethylene, polypropylene, polystyrene, and polyamide. Additional plastic collected directly from the river with unknown submerged duration were also included. After the incubation period, several measurements were carried out. This included chlorophyll A, 16S and 18S sequencing, dissolved oxygen, ecosystem production represented by NEP and respiration, as well as nutrient change involving nitrogen, phosphorus, and dissolved organic carbon. This combination of measurements allowed the researchers to evaluate both microbial development and ecosystem response. Now, let's look at what the researchers found. First, photosynthetic biomass remained low across plastic surface with a mean value of 0.13 plus minus 0.18 microgram per square centimeter. Next, bacterial communities were mainly dominated by Protobacteria and Actinobacteria, while algal communities were mainly represented by Ochrophyta and Chlorophyta. The functional analysis also showed dominance of chemoheterotrophic and aerobic organisms, including pathway related to nitrogen cycling and hydrocarbon degradation. Another important findings was that biofoil plastic significantly reduced dissolved oxygen and resulted in more negative NEP values. This indicates net heterotrophic conditions. So overall, the presence of plastic was associated with change in microbial activity and ecosystem process. At this point, we can notice something interesting. Even though the study focused on plastic, the impact was not limited to the physical presence of plastic alone. The microbial communities developing on those surfaces appeared to influence oxygen dynamics and ecosystem metabolism. So, rather than acting only as physical waste, the presence of plastic was associated with change in microbial activity and ecosystem process. Based on this finding, low photosynthetic biomass suggests limited photosynthetic activity occurring on plastic surface. At the same time, the dominance of bacterial communities together with chemoheterotrophic and aerobic function suggests active microbial metabolism. The reduction in dissolved oxygen and more negative NEP values indicate change in ecosystem metabolism associated with biofuel plastic. This finding suggests that microbial activity developing on plastic surface may influence ecosystem functioning. If we connect this back to the introduction, earlier we discussed how plastisphere communities may influence biodiversity, nutrient cycling, oxygen dynamics, and productivity. Based on the findings presented, these effects can be observed through change in microbial activity and dissolved oxygen conditions. Taken together, these findings suggest that microbial activity developing on plastic surface may influence ecosystem functioning. Before moving to the final point, I would like everyone to think about this. When we see plastic floating in rivers, we usually focus on what is visible. But based on this study, there may also be invisible biological change happening on those plastic surface. So, to conclude, plastic surface supported microbial colonization in large rivers. These plastisphere communities were mainly dominated by heterotrophic microbial function. In addition, biofuel plastic altered dissolved oxygen dynamics and ecosystem production. Overall, this study demonstrates that plastic pollution may influence freshwater ecosystem process. For the acknowledgement, lastly, the authors would like to thank the editor and Animamus reviewers for their valuable comments. The author also acknowledged the Department of Earth and Environmental Science and the Interdepartmental Network of Spectroscopy at the University of Milano Bicocca for providing Raman facilities. Before I end, I would like to leave everyone with one final thought. If something as common as plastic can influence microbial communities and ecosystem process, what could happen as plastic pollution continues to increase in freshwater system? So, that is something worth thinking about as plastic pollution continues to increase globally. Thank you for your attention. I welcome any question.